Hello, everyone. I hope you're all staying safe and healthy. My name is Jason Drenning. I'm Vice President of Business Development and HEOR at Staten Med Research. I have a background in HEOR on the manufacturing side, as well as with various HEOR consulting shops, including experience designing studies, working strategically with pharmaceutical medical device partners in determining study needs, research ideas, as well as business strategy, including external control arms and historical control arms. With me is my colleague, Nora Janjan. Nora?
Thank you, Jason. I was a tenured professor at MD Anderson Cancer Center, where I served as vice chair of the Institutional Review Board and held a dual appointment in radiation oncology and symptom research. Based on this experience, I served as a member of the NCI Central IRB for National Cooperative Group Trials. I also was selected to be a member of the Medicare Coverage Advisory Committee, and I'm a past president of ASTRO. I'm one of the few physicians nationally that has a master's in public service administration and an MBA.
Thanks, Nora. And to be clear, Nora is our Chief Medical Officer at Staten Med. Staten Med's an HEOR consulting company, and really the purpose of this video is the first in an introductory video to complement a series of other videos exploring the topic of synthetic control arms, historical control arms, external control arms. For the purpose of this particular presentation, we're going to refer to them as historical control arms. So we're seeing a significant uptick in the number of these studies being conducted, various reasons behind that, but we want to talk through this a bit, particularly with Dr. Janjan, because of some of the experience she's had at MD Anderson. You know, the recent increased popularity of this approach, Nora, is a little bit deceiving. It's not necessarily an innovation. Historical controls have been around for a while. So maybe talk a little bit about your experience with historical control arms at MD Anderson specifically.
Jason, thank you. You know, from its beginning, MD Anderson conducted sequential clinical trials using the patients from the previous clinical trials or demographically matched patients who received the existing standard of care as its historical controls. And this approach that borrowed control patients achieved a sufficient statistical power. It reduced type 1 error and required a smaller investigational patient cohort, all of which increased the efficiency of our cancer research. And importantly, all of the patients within the clinical trial had access to the novel treatment rather than being randomized to a potentially inferior standard of care. Using a dynamic versus a static borrowing for the historical control arms, MD Anderson was able to make rapid progress in clinical trials using sound statistical methods.
That's interesting. So I think, you know, one of the questions that arises as we talk to folks, particularly in the pharmaceutical space, as they're thinking about an external control or historical control as an option, revolves around some of the endpoints and whether or not those can be matched and how to find patients and matching those patients in the control cohort against those in the prospective study arm. So for these types of studies, what specific endpoints are being measured?
Well, MD Anderson has been around a long time and they have a vast number of patients and a lot of rich data. They've also published many retrospective reviews to demonstrate therapeutic trends and outcomes. And this was especially important to identify long-term therapeutic success and the sequelae like second malignancies developing almost 2 decades later in the radiated field. This is in contrast to many prospective randomized controlled studies that often had very different endpoints and much more limited follow-up. Cancer research evolved. MD Anderson transitioned its research approach to prospective randomized clinical trials along with the other major academic medical centers around the country and also because of the requirements of regulatory agencies. So while there were great discoveries made during this time, There was wide concern from many stakeholders that randomized controlled trials delayed the introduction of innovative and potentially life-saving therapies to the clinic. And the controversy that surrounded Gleevec is a case in point. Bottom line is randomized controlled trials just couldn't keep up with the pace of rapid scientific discoveries.
Yeah, I think that makes sense. I think what's interesting is there's certainly resistance to an approach that's not the gold standard of the randomized controlled trial. I think the challenge is proponents of randomized controlled trials that oppose these synthetic control arm, historical control arms, maybe aren't considering some of the shortcomings of randomized controlled trials. So there certainly are challenges that come with those types of studies that don't get brought up, I think, enough. So what are some of the challenges from your perspective, Nora, as a clinician with prospective randomized controlled trials?
Well, as a clinician, there are two major drawbacks of a randomized controlled study. And one are the large numbers of patients required to conduct a study. And often when you have you're dealing with diseases that are rare diseases, and it's hard to get patient accession. This is an impossible task. Even though that randomized controlled trials have been considered the gold standard, it's oftentimes very difficult. It takes years to acquire the large patient populations to provide sufficient statistical power to the study. And only 50 to 75% of patients are receiving the innovative therapeutic therapy. So standard of care is administered to the control arm. So study and control arms, having both study and control arms, a prospective randomized controlled trial essentially takes twice as many patients to complete. The second and very important consequence of requiring large numbers of patients is slow patient accrual. And strict patient eligibility also is part of that. And that can slow patient accrual to a randomized controlled trial and may not reflect real-world treatment populations and practices. And as less than 5% of cancer patients participate in clinical trials, getting innovative therapies to clinic is even slower. So these two factors in particular really slow down innovation getting to the clinic.
Well, I think in addition to the cost and speed to market and speed to therapy, there certainly is a patient ethics component of this too that is something that needs to be discussed. But certainly from what we're hearing and seeing, historical control arms could be the future, especially for rare diseases in oncology. So Nora, your thoughts on that? Well,
they're not my thoughts. They're Dr. Janet Woodcock's perspective on it. She, as director of the U.S. Food and Drug Administration, the FDA Center for Drug Evaluation and Research, she's acknowledged that the current drug approval system is broken, and there needs to be new ways of collecting and utilizing patient data. And the emergence of big data provides new opportunities to efficiently conduct large-scale studies that have many of the benefits of randomized controlled trials while being generalizable to broad populations. And in fact, a lot of the concerns of patient bias can be eliminated through current big data techniques. Big data studies reduce the time to study completion and the time to regulatory approval, but most importantly, the time it takes for patients to have access to the therapeutic advancement. Although randomized controlled trials are considered the gold standard, the role of historical control arms is expanding. Historical control arms in oncology improve the efficiency of the study, but also inject real-world data and real-world evidence in the clinical trials. And through FDA recommendations, formal acceptance of historical control arms was codified in the 21st Century Cures Act, signed on December 13, 2016. This act indicates the FDA will accept real-world evidence in regulatory decisions for oncology and rare diseases. It's a formal acknowledgement of historical control arms, and it also recognizes that statistical designs of clinical trials must be more nimble and account for the limitations of patient populations and the ethics of receiving a standard treatment that's of limited benefit.
Makes sense. Nora, this is extremely useful, and certainly your experience at MD Anderson is very relevant to this topic. We've covered a lot in a short amount of time. So just to kind of circle back, a few take-home messages that you'd like to share.
Well, now that historical or synthetic control arms are formally accepted, and there are many advantages in clinical trials, including serving as a control arm and studying rare diseases and in oncology. It reduces the total number of patients, allows more rapid completion of clinical trials, and that cascades into more rapid regulatory approval and patient access to therapeutic breakthroughs. It reduces the economic costs of developing new novel therapeutics. It overcomes the ethical considerations of randomization in trials with standard therapies of limited benefit. And for studies needing long-term follow-up, a control arm population can be expanded and can be sustained to retain statistical power.
Great. Thanks, Nora. So this has been really helpful. We will be continuing to develop these videos. Our next topic will cover the impact of COVID-19, and in particular, the potential reduction in prospective trial enrollment and some of the hindrances that COVID is creating, particularly in terms of cancer trials. Thanks a lot, Nora.
Jason, it's been my pleasure.